most of us could live to 90, 95 in good health. I mean, just imagine in comparison to what we have, where most people live in good health until 65. If these interventions could be implemented on a population basis, it would be an absolutely tsunami in terms of change in life expectancy.
Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Dr. Gil Blander. Welcome to Longevity by Design, how to live a longer, healthier life. We are produced by InstaTracker, your science-based guide to optimize your body from the inside out. Today on Longevity by Design, we are honored to welcome Dr. Verdin, who is a world-renowned neuroscientist and president and CEO of the Bach Institute of Research of Aging, the first independent U.S. research center dedicated exclusively to the biology of aging. Trained as a physician at the University of Leeds and Harvard Medical School, Dr. Verdin has held key academic and leadership roles at the NIH, UCSF, and the Gladstone Institute. His research focuses on how epigenetic regulators such as serotonin and histone deacetylases and metabolic signal like NAD+ and ketone bodies influence immune aging, inflammation, and age-related diseases, including Alzheimer's. With over 210 publications and numerous patents, he brings cutting-edge science with translation innovation, including clinical trials like the Bach Institute ketone ester study. Eric was a guest at this podcast twice already, and in this episode of Longevity by Design, we will discuss Eric's recent exciting paper published in the journal Aging Cells a couple of months ago, which explored the effect of therapeutic plasma exchange on biological age. Welcome, Eric.
Welcome. Delighted to be here. Thank you for having me.
Yeah, I'm very excited about this paper. And when I read it, I said we have to have an interview about it because, as we discussed before recording, Eric, there are a lot of recommendations of a supplement or intervention that are not backed by clinical studies. So when I've seen you and your team conducting a clinical study on the therapeutic plasma exchange, I said we have to discuss it in this podcast. So I would like to start with the background and basically why have you decided to study TPE? What was the reason of running this study, and what was known about the effect of therapeutic plasma exchange on health and longevity before this study?
So the background of the study comes back a long time ago, and I'm sure you have heard about Heterochronic Parabiosis. It's a mouthful of a term, but what it indicates is experiments that were conducted a long time ago in which you can essentially, it sounds a bit gruesome, but that's the experiment, you can stitch two mice together via their skin. When you do this, what happens is after a few weeks, the mouse actually generate a unique circulatory system. That is, the capillaries will exchange blood between the two mice. And so what this experiment allows you to do is to do this with mice of two different ages. And this is work that was pioneered by Tom Rando at Stanford, where they were able to show that doing so has effects both on the young mouse and the old mouse in the pair, in that the young mouse appears to get older faster, and the old mouse appears to be rejuvenated. And so this generated an enormous amount of interest in the field because it indicated that there are factors circulating in blood that might have positive effect. And so it has generated a whole cottage industries and new companies looking for factors that were present in the young mouse that could actually rejuvenate an old mouse. And we've all heard about GDF and other factors that have been identified. Tony Wiscoré was involved in this also, showing that you can actually have protective effect on the brain, which generated obviously a lot of interest. It also generated a whole side in the industry of people proposing transfusion of young blood to older mice or older people, which obviously is something that was shut down by the FDA for obvious reasons, because transfusions carry a whole series of inherent risk. So that initial group of work sort of was based on the idea that there was a factor in the young mouse rejuvenating the old one. But what was not paid attention as much was the idea that providing a new system, a new regulatory system to the old mouse might allow it to subtract factors that were detrimental. And so this experiment, this hypothesis, so the idea that in the hybrid mouse, you were sort of diluting a series of negative factors in the old mouse, was not really paid attention enough, I think, at the beginning. But eventually, Irina Conbo and her colleagues at Berkeley conducted a simple experiment, which consisted in withdrawing blood from an old mouse and reinfusing the cells, so there's no cytopenia. And they found that, remarkably, these mice actually benefited significantly in terms of their health span and lifespan. So that generated a whole series of secondary experiments suggesting that both mechanisms are likely to be playing. When you do heterochronic parabiosis, there is addition of rejuvenating factor and there's subtraction of factors that are promoting aging. And so the beauty is that there is a technique in human called therapeutic plasma exchange, which does exactly this. Is a medically approved procedure with centers around the country that will withdraw blood from one arm. It goes into a machine where the cells are centrifuged, separated from the plasma. The plasma is discarded, and then the cells are reinfused along with fresh solution. The reasons why this is used in a clinical world is one of the indications. There are multiple, by the way, is, for example, some autoimmune disease where you might have a pathogenic antibody that you want to remove. You can actually do this, and this is a successful and approved treatment for a number of conditions. So this yielded the idea maybe we could actually test in humans the hypothesis that doing so, TPE, therapeutic plasma exchange in humans, might actually yield some degree of beneficial effects against aging. And I want to amplify the point that you made at the beginning of our presentation. I'm a really strong proponent of the idea that our field will establish its legitimacy when we abide by the highest standards of demonstration of the efficacy of what we propose. And as you know, there's a proliferation on Instagram and other places of treatment and vitamins and supplements and so on, interventions, almost all of them unproven. And so I think as a field, if we want to establish the validity of what we do and be taken seriously, I think it's incumbent upon us to really apply those rigorous standards. And so the whole idea of doing this was a company that was initiated in collaboration with W. Kiproff, with funding with Khosla Venture, to actually conduct a rigorous, double-blind, placebo-controlled, which we can talk about this later, a trial that would test the effect of TPE on patients. And so to do this, we applied all of the tools of the Omics and clinical measurements that we could do, and this resulted in the clinical trial and the paper.
Yeah, thank you so much for the great introduction to this field, because it's a pretty complex, and I'm sure that our listener will appreciate the detailed background. So thank you so much for that, Eric. And if we are, you said the method, so if we will try to dive into that, in the trial you include either biweekly therapeutic plasma exchange or biweekly therapeutic plasma exchange plus immunoglobulin, or monthly therapeutic plasma exchange, or a placebo arm. Can you explain the rationale for this specific regime and why it was a single blind setup and not a double blind?
Yeah, double blind. I mentioned double blind. You're correct. It was single blind. It's almost impossible to do. You have to have someone set up the system. The patients certainly did not know in terms of what was happening, but blood was withdrawn and was reintroduced, so that there was essentially a true placebo control, which, by the way, has been very rarely conducted in this type of intervention because it's obviously not easy to do. Now, the reason for the nature, the different forms of intervention that we conducted is we started from the standpoint that we were hoping it was going to be beneficial. The question is, and I want to highlight this is not a completely benign intervention. It involves, withdrawing significant amount of blood, circulating the blood through a machine. So we were concerned that, we were trying to address three major questions. One, could we actually demonstrate any effect on any variables? And we started really, frankly, open to the idea that we might not see something. So we went pretty wide in terms of all the things that we measured, all of the omics, clinical parameters, and so on. The second one is we did not know how frequently you would have to do this. And these are still, partially unresolved questions. Obviously, because of the nature of the intervention, it's not cheap. It involves the patients coming to a clinic, sitting in a bed for 2-3 hours. So this was not something benign. So we were concerned that not to do enough and to do too much with both. So that's why we tested two different protocols. Another variables that we still do not know is once you have initiated the therapeutics and you stop, how long does the signature remain? That is it something that people will have to do once every 6 months or once a year, or a series of 3 followed by an absence for a year? So this is really what all of the questions that we're trying to address. And our goal here is not only to create a company. You are a businessman, so you understand the imperatives of having a business that generate revenues and is ultimately successful. Our goal really was to understand how can we benefit those patients and what is the optimal, most cost-effective manner for this treatment to be administered with maximal result.
Yeah.
And I should say, this is an ongoing process. Obviously, this was the first study. We published it because we obtained some positive results very quickly. I was anticipating that we were going to have to conduct 2 or 3 trials to actually get to this point. The fact that we got a signature right from the beginning was extremely encouraging and really led to the study.
Yeah, and the N number was pretty small. So again, it showed that it's a significant result that went way above the noise of the background, which is very exciting, and very exciting, at least to me. And I think that there might be a lot of implication in the future. You alluded to that, and you said that you collected also a lot of multi-omic variable like epigenome, proteome, metabolome, glycome, cytokines, and so on and so forth. What was the biggest challenge in integrating such heterogeneous databases and collecting so much data?
Well, maybe I can say a few words about the whole idea of phenome health and the whole idea of these omics. I think it really underlies a lot of what we do here at the Buck, and I think will underlie what we call the future of precision medicine. This is the idea that if you look at the way medicine operates right now, it relies on a really small number of variables. Your typical blood panel that you have annually at your doctor will cover maybe 100 biomarkers at most. And this is in contrast to the amount of data that we can generate using the most modern technologies. If I do an RNA-seq analysis on your blood, we can measure probably 10 to 15,000 transcripts. If I go to proteomics, we can identify 6,000 to 7,000 proteins. If we go to metabolomics, we can identify another 5,000 metabolites. So we have, I would say, an embarrassment of riches at this point. And there's this incredible discrepancy between what we measure actually to make clinical decisions on patients or to assess the efficacy of drugs and the amazing amount of technology that we have. So our long-term, and I didn't even discuss wearables and all the data they generate, medical clinical record, and so on. So there is this incredible discrepancy between the two, and much of our work at the Buck is really to usher in this new era, what we call precision medicine. And so, as you know, we've recruited Lee Hood and Nathan Price, who are, along with Phenome Health, this is Lee Hood's organization, are really pushing the boundaries in terms of using these most comprehensive tools to dissect human biology and hopefully the goal of all of this is to essentially identify novel biomarkers of aging. I mean, we have the whole epigenetic clocks. Everybody's heard about those, but they are just the tip of the iceberg. Tony Whisperay just came up with this beautiful organ-specific clock, which he's commercializing through a company called Vero. There's so much happening in the field. Facial clocks, skin clocks. I mean, the list goes on and on. So the question, one of the central questions to all of this is, if you generate this type of data, this is not the data that you and I or any physician can look at and try to make sense of. And so different areas that we are investigating is first trying to establish, of all these variables, what correlates with what. How can we make sense of this enormous array of data? So the idea is to take all of the data and project them onto what we call knowledge graphs, which are essentially sort of network made up of nodes that comprise all of these variables, and then try to see where are the correlation, what is predictive of what. And so obviously this is a type of work that requires artificial intelligence, and much of our effort, along with Lee and Nathan, John Newman, and others at the Buck, is really focused on really deciphering this new biology. And you might have heard about the Prosper grant application that ARPA-H is currently offering. And so we have several applications going on really at the cutting edge of biomarker research, trying to really usher in this new area.
Yeah, no, and we interview Lee Hood and Nathan Price in this podcast when they published their book, and definitely they are in the cutting edge of the technology. One point that you mentioned about your primary care physician. So you said 100 biomarker. I think that you are pretty optimistic. When we looked at that, it's on average is around 18. So the physician only tests around 18. And I agree with you that collecting a lot of data and then trying to understand what is meaningful and what is not, and then hopefully include it and make it 19 or 20, but add something that is very meaningful. That's the future, and that's very interesting. Good that organizations such the Buck are doing that. So congrats for that. My next question is about the epigenetic clocks that you used. You basically looked at 36 different epigenetic clocks. How did you choose them? Or maybe you just use every clock that's available and then use all of them? Can you elaborate on that?
Yes, we did not choose them. And I was struck by the fact that, I'm as excited as anybody by what the epigenetic clock and the pioneering work of Steve Horvath have brought to the field of aging, because, there was a period, you might recall, back in the 80s and 90s where the NIA proposed a large number of studies identifying biomarkers of aging, which came back with really not much. And there was general consensus in the field that biomarkers of aging do not exist, or they're very hard to discover, except for a few variables. What Steve Horvath's work demonstrated in the most elegant manner was the fact that even using a simple marker as epigenetics, you can actually identify really strong predictive marker. That being said, Steve's original paper was followed up by a whole series of variations on this. First, clocks that measure just chronological age, then biological age, then clocks that were predictive of disease, rate of aging, such as Dunedin Pace by Dan Belsky and his colleagues. So we're now have an embarrassment of riches. We have so many of these clocks. Nobody knows what to do with them, at least from the clinical standpoint, where the criteria for introducing such markers are extremely high. And so the other problem is, strictly from a personal point of view, as I do a lot of self-intervention, I do measure myself as much as I can. At one point, I conducted sort of an analysis of all of these clocks on myself, and my age varied from 25, and I just want to reassure the audience, I'm not delusional. I'm not 25, I don't look 25, and I certainly don't feel 25, but one of these clocks repetitively gives me at 25. Another one gave me at 68, which is my true chronological age. So I varied between 25 and 68, and with every number in between. And so this really gave me pause in terms of whether these markers are ready for prime time in their current form. which, by the way, is a subject of work here at the Buck and many other places, trying to understand what are these clocks measuring and how do we use them, how do we deploy them clinically. So based on all of this, we started with the assumption that none of these clocks really tells an exact truth. One thing that has emerged, though, is that many of these clocks appear relatively stable over time, and they might be speaking to different aspects of biology. So the question was, in the longitudinal study, the way we conducted, we thought if any clock gave us a signature that changed as a result of the intervention, this might be a clock that we should use to follow these patients. And that's exactly what we did. So we started unbiased. And I mean, frankly, at the end, as you have seen in the paper, the clocks were all over the place, but there was a clear signature that globally, and not all of them, I should precise, were moving in the right direction after a unique set of intervention. And we thought, okay, this is something to share with the community and for people to chew on and try to make sense of it. And I think this is essentially what the data showed. I mean, on average, I believe we had a 2.5-year reversal averaging these clocks. Some of the clocks actually move much more significantly. So again, this is a reflection of our lack of understanding of what these clocks are really measuring, especially since they are all relying on blood. All the ones we use were relying on peripheral blood. And remember, this is the major compartment that we were targeting with this intervention.
Yeah. And I think that you raise a very good point. It doesn't matter if the clock telling you that you are 25 or 68. The bottom line is what happened, what is the delta? And
Exactly.
the most important is the delta, because as we know, those clocks are not very accurate to predict your age. But if the delta is similar, as you said, as you showed in the paper, that means that indeed the therapeutic plasma change have some effect. And it sounds like in most of those clocks, it was a positive. It decreased the age. So
Exactly.
that's an interesting point. And if we are talking about that, so the biggest effect was with a therapeutic plasma exchange plus immunoglobulin. As you said, it's decreased the age, if to be precise, around 2.61 years.
Yes.
And then that was at 1 month. And if you done just a therapeutic plasma exchange alone, it was around half of it, like 1.32 years effect. In your opinion, what is the clinically meaningful of this value? And what are your thoughts about the durability of the results?
As a Longevity by Design podcast listener, you understand the value of improving your health for today and for all the years ahead. And if you want to live your healthiest, longest life possible, you need to understand what's going on inside. At Inside Tracker, we take a personalized approach to Healthspan optimization that eliminates guesswork from your wellness plan. Inside Tracker analyzes blood biomarker and DNA data, along with physiomarker data from fitness trackers like Oura Ring, to deliver personalized food, supplement, lifestyle, and exercise recommendations that allow you to take control and improve your health span. And for a limited time, Longevity by Design listeners can get 20% off at the Inside Tracker store. So if you're ready to receive a personal health analysis and data-driven wellness plan to optimize your body for the long haul, then it's time to start inside. Visit insidetracker.com/podcast to get started today. That's insidetracker.com/podcast to get started today.
So you make a really important point. So two important points, and I'll talk about both. One, the frequency or the number of times that you repeat the intervention. And the second one, the possible negative effect of the intervention. And the second point, I mean to say the immunoglobulin, because this is something that we still do not understand. It turns out that the patients in which we saw the biggest signature were those who we were all reinfusing a saline type of solution without human albumin. Some patients received a dose of immunoglobulin and some didn't. The reason we gave the immunoglobulin was when you get rid of the plasma, you get rid of immunoglobulin. These are the antibodies that we use to fight infections. Mm-hmm. And so the concern was we're going to be depleting people of critical antibodies. And remember, when the study was initiated, we were at the end of the pandemic. And I frankly was concerned that we were going to make people partially immunodeficient for a period by removing their vaccination antibodies, and so that people might get more COVID, which would not have been the intended action. Now, it turns out it's not only COVID antibodies, it's also influenza antibody. It's antibodies against a whole series of viruses that we know can be very harmful, especially in an older cohort, the way we conducted the experiment. So the idea of giving the immunoglobulin back was to try, this is a hyperimmune immunoglobulin solution that you can actually purchase. And it's been shown in other systems to actually be conferring some resistance to some viral infections. So this was not just giving a protein, this was giving a protein that we thought was important biologically. It turns out that that group of people actually benefited the most from the procedure. It turns out also, as we're learning, that injecting hyperimmune immunoglobulin can have modulating effect on the aging process. And this is something, a question, results that was unexpected, that brings up a whole series of interesting biology. Is there something that could be done beyond TPE, just giving immunoglobulin?
Yeah.
So that's an interesting question. The second one was the frequency. As I mentioned, this is not a totally benign procedure. I mean, it's not toxic, but it's medically involved, including being hooked up with 2 big needles and so on. So the question was, we have a saying in French that, the enemy of good is better. And so, of course, I'm maybe doing it 10 times would have been better than doing 5 times, where we were concerned that there was going to be some degree of burden associated with the procedure. And that's exactly what we saw. And so here we saw people who received 1 actually benefited more than people who did 2. Now, I should add to this that this is only a hint. We need to do more work to really understand the best frequency at which we get the maximum efficiency, so the maximum efficacy. And this is where pretty much the field is right now.
Yeah. And another interesting observation is that the benefit diminish after multiple sessions. So basically, and I think that you mentioned that maybe you don't need to do it many times, but also even if you do it many times, you don't see the benefit. So how do you explain that basically it's diminished after a while?
The short answer is we don't know, but it raises a very important question in terms of the frequency at which we're going to have to be doing this. To be sure, we did not address that question, so we have not measured how long the response lasts. What happens if we take these patients who showed this great response and follow them 6 months, at 1 year, or 2 years? When is the next time where the response has diminished? Does it diminish? I don't know if this reversal is permanent or does it go away. So I would predict that after a while, the forces of biology will come again, and the response will be erased, and this would be the time at which you would want to do another one. Yeah. So this is where we are at this point, is really trying to decide what is the optimal frequency. Should you do 1 or 2 or 3? And when you've done so, how often do you repeat it, basically, after 6 months or after every year? I can imagine a system where people would have one of these procedures done once a year and then come back the year after, and this would result in the optimal effect that we can measure.
Yeah. And another exciting result, and I assume that maybe it was part of the work of Lee Hood and Nathan Price, is that you found some signature of biomarkers such as bilirubin, glucose, and liver enzyme that actually can predict who is going to respond better and who won't respond. So I'm curious to know, how did this integrate into your prediction model, and how strong were their predictive effect?
So this work was done by David Furman, a faculty member and rising star at the Buck Institute, who also leads our bioinformatic efforts here. And David, as you know, has been interested in immune aging for many years, part of the 1000 Immunome Project in collaboration with Mark Davis at Stanford. And so really knows the whole idea of immune aging in humans probably better than anyone else, and really played a key role in helping us to analyze these data. Now, one of the things that we were trying to do in these experiments, since we measured people before the procedure took place, was to identify biomarkers that will be predictive of who responded and who did not respond. And to be sure, some people did not respond as well as others, and some actually showed very dramatic effects. So the question that was asked is, in those markers that were identified early before the interventions, which one were the most predictive of a positive response? And this is the ones that you just mentioned. Now, what I cannot tell you yet is how does this relate to the procedure, to the biology, and so on. So it could be that, for example, someone who shows, one aspect of heterochronic parabiosis that was not this sort of considered beforehand, as I mentioned at the beginning, is the idea that an old mouse benefits from being stitched up to a young mouse because all of a sudden it has a new liver, which we know, plays a key role in detoxification. It has a new spleen, which we know plays a role in sort of filtering the blood and all blood cells. So you're essentially providing a whole new filtering system, which is what we do with the machine. So the fact that some liver enzymes might be correlated with this could be an indication, for example, that maybe those patients that had more liver aging, again, this is something that we now know we learn how to do, is to measure organ-specific aging. I mentioned Vero, that's one company that's doing this. You can now, we could predict maybe those patients that had less liver function actually benefited more. And I think this is, what we're exploring right now is what is all of this data telling us, who will benefit from the therapy. I'm not sure, frankly, that everyone benefits, and we want to be sure that we apply this technology to the people who are going to see the biggest effect.
Yeah. And another surprising result, at least for me, is that the side effect or the safety profile was pretty good, and only 2 subjects discontinues the experiment, and only 1 related to adverse effect. What do you see about that? And were you expecting that, or were you expecting much more discontinuous based on the historical result?
That's a great point. And that's one that I would highlight as one that your audience should be concerned with. TPE in the right hand, as in ours, and this was pioneered and really greatly contributed to by Dobry Kiprov, who was a co-founder of the company. By the way, the company has now been renamed Circulate, and Dobry Kiprov is no longer associated with it. But the Dobry has established extremely high standards in terms of safety for the procedure. And I would argue that not all clinics that do TPE actually have the same safety profile. There's quite a few have shown in the past, significant side effects. So we still benefit from everything we've learned from Dobry in terms of optimizing the conditions so that a very small fraction of people experience any kind of side effects or problems. And I should say this is not true of all clinics. So buyers beware. Before you let people do TPE on you, make sure this is an accredited organization with the highest standards. And I would argue ask for, the rate of complication, which should be existent because it is not negligible at some sites.
Yeah, I know, I agree, and I think that, and hopefully we'll discuss it later if we'll have time. I think that, in my opinion, the end goal is not all of us doing TPE, but a different way that we'll discuss later. But I want to discuss about more like longer term and more like physiological outcome. So your study look at some molecular and age shifts, but haven't looked at more like functional outcomes, such as cognition. Maybe you've done it, but it wasn't published. Physical performance and so on. Are you planning to look into that in the follow-up study?
Yes, I hope so. You're referring to the whole idea of intrinsic capacity here, which I think is a concept that was initially introduced by WHO as a way to measure aging. It's one thing to measure aging in people by looking at parameters such as the Omics and all this. It yields a lot of data, but it is meaningless to the patients. The WHO came up with the concept of intrinsic capacity, which is a series of metrics that rely more on really what you feel as you age. And obviously our goal as we are fighting aging is to bring people in the highest state of emotional, physical, capacity as they age. People do not care about living older if it means being in a wheelchair or not being able to read a newspaper or to converse with friends. So this WHO intrinsic capacity concept is a way to try to address this. So it's based on a whole series of metrics linked, for example, to mental health, linked to physical agility, physical mobility, depression. So all kinds of variables that we can now quantify and really add a whole dimension to the biological markers. And I should point to a really important paper that was published by David Furman, again, our colleague here at the Buck, where he actually joined these two worlds, the world of epigenetic clocks and the world of intrinsic capacity. And he, interestingly and remarkably, I don't know if I would have believed it would have worked, but it does work. You can actually identify epigenetic markers that correlate with intrinsic capacity. And so this is a new clock that David has generated. And obviously our goal and our interest is to go back and revisit some of the samples that we have and expand on the findings that were present in this initial paper.
Yeah, and I just want to follow on David Foreman. We interviewed him in the podcast a couple of years ago, and I have to say that looking at what's happening now in the aging-related research domain, David is one of the most productive scientists. He's publishing a lot of paper and a lot of very good science. So definitely,
we're very proud of him.
Yeah.
And I think this is the greatest joy I have in this job, to be honest, is to see our junior or mid-level faculty emerge as leaders in the field and continue, the long tradition that we've had for a number of years with Judy Campisi, now with Nathan Price, Lee Hood. David is certainly an upcoming star. So it's actually, it's exciting. It's what gets me up every morning to come and work here and make sure the Institute prospers.
Yeah, no, so congrats for that. Definitely David's work is very impressive. Another follow-up question is about combinational strategies. So you use TP and immunoglobulin, and it's interesting that it's working, but do you think that it might make sense to combine it with a diet, exercise, analytic, or any other approach, and to see whether there is an additive effect or not, or it's not something that you are thinking about?
Well, you probably know this, but this is the thing that's closest to my heart as a physician, is the whole idea as we move this field forward with the aim of increasing lifespan and health span for everyone, is that we do know the field has identified a whole series of interventions that result in a very large increase in health span and lifespan. And so these are the lifestyle factors. I probably not need to mention them to this audience, but it's nutrition, it's physical activity, it is human connection, stress mitigation, sleep, all of the things that your mom or your grandma told you and that your doctors probably ought to tell you. So one of the sort of nightmarish version is that of our work would be that all of these factors that we know can lead you to lead a much longer and much more healthy life, based on what we know today. And this is not really uniformly appreciated. Most of us could live to 90, 95 in good health. I mean, just imagine in comparison to what we have, where most people live in good health until 65. If these interventions could be implemented on a population basis, it would be an absolutely tsunami in terms of change in life expectancy. And, quite often people misunderstand what I'm trying to say by thinking that I'm all about health span. There's nothing further from the truth. I mean, we study longevity at the Buck. I'm passionate about the science and animal models, but I also think that there is easy gain that can be made on a population basis based on these lifestyle factors. And the reason why I push for them is that I tell people this is the best way you will benefit from the future discoveries of the field and stay as healthy as you can, because the drugs that target aging are coming and you want to be able to benefit from them. So I really view our field as being made of two aspects. What can we do now? And we know what we should be doing. And tragically enough, we're not doing it as a country. So that's one aspect. The second one is, how do we push the envelope once we have optimized people to 90, 95 in good health? What are the next steps? And this is where procedures like TPE, other factors can play a role. I want to say make another point, which is that we know that we all have different risk factors for disease. And some of us, simply based on our genetic background, age faster than others. So I can imagine a day when we will be able to identify these people with a fast aging protocol, sort of a phenotype. And really, those would be the first one to benefit from this, frankly, and the ones where we can get the maximum effect. Now, what role exactly will TPE play in the whole armamentarium of interventions that we have in the future? I do not know. What combination will be most effective? I do not know either. This is the work that we're doing, and this is what keeps us going, trying to make discoveries and to move the field forward.
Yeah. And Eric, you made a very important point that I want to repeat it for our audience. So changing your lifestyle, if you do everything well versus if you do everything not well. So basically, if you smoke and don't exercise and eat like a pig and they don't sleep well and don't have a social relationship and so on and so forth, versus someone that's doing everything well, there is a lot of data that show that you can live up to 20 years longer, and your health span will be 20 years longer. So it's a lot of years.
And it's a lot of years. And, what I tell people also personally, when I got this job, I think I've always tried to be healthy, but I would not have called myself a health freak. Yeah. Maybe I'm more of one now because I've always felt that, given the position I have at the BAC, in some way, I don't speak for the whole field, but I'm a strong voice in the field. I want to be able to share with patients and people what I do know, and I want to be able to speak about it from a personal perspective, which is also important for people. And so when I took this job 9 years ago, the last 9 years, I have systematically explored a whole series of interventions, and I go from rapamycin to metformin and so on. And so that I can speak to people about personal experience. One of the most important things that I've discovered in this process is the fact that once you optimize your lifespan or your lifestyle, you actually benefit in the moment. And I've stopped thinking about how long I'm going to live because, frankly, it's out of my control, at least a big part of it. But I can guarantee you that one of the biggest discoveries I've made is the quality of my life on a daily basis has increased immeasurably. And I tell people, at age 68, I feel better than I have my whole entire life. I don't have any pain anywhere. I exercise an hour every day. I try to eat reasonably. I'm not a saint, so I certainly don't advocate for this. And all of this really has increased my quality of life in an incredible manner. And you can speak to many people who have gone into this journey, as I suspect you have too. It's black and white. It really is a big difference in terms of the quality of your life. And that's already good enough, at least at some level, for me at this moment. And whatever happens in the future, I think it's partially out of my control. But I know that I'm living the best life that I can possibly live.
Yeah, absolutely. And I think that another good point that you mentioned is if our audience or the people that listening, you will do that, you have a better chance to get ready when to be around when those new kind of treatment will come and hopefully will be validated by institute like the BAC and will be ready for the mass market, such as ATP, cellular reprogramming, and other. So you have basically a double incentive in a way. First, you live better now, you feel better. Also, you most likely will live longer, but also you have the chance to be around when the life-changing intervention will come and will be able to cross the glass ceiling of 122 that nobody else that we know across it. So I'm 100% with you with that, and thank you so much for explaining it so well. I have another question, maybe the last question about it, the TPE. How do you see the future of that? Do you see all of us going to a clinic and connecting ourselves to a needle and receiving a session? or more like isolating, as you mentioned at the beginning, the right component of the TPE and then taking a pill once a day, and that's it. What is your vision?
Now, clearly, I don't think this is something that's going to come in a pill, to be honest, because this is a subtraction procedure. So this is one in which we're removing a whole series of factors. Now, one possible future direction that you could imagine, and we don't have any data on this, I'm just trying to imagine what this could be. It could be that when we're removing the whole plasma, there are 2 or 3 or 4 unique molecules that exert a toxic effect that are not being removed efficiently, say, by your liver. You can imagine if such compounds or molecules, for example, thinking about TNF as a factor, the pro-inflammatory cytokines that we know appears to be linked with aging. It could be that removing that one alone would be sufficient.
Yeah.
Imagine also a world in which you would identify 4 or 5 of these molecules, and you would generate now a column that you could filtrate your blood with to eliminate just those 5 molecules. It could be that you might even see a bigger effect. So there are, during aging, there's a lack of positive factors that contribute to rejuvenation and maintaining a youthful state. But there's also the progressive accumulation of a number of factors that we know are toxic. I mean, that's been clearly documented. And there's some evidence that some of the approved drugs actually are targeting IL-1 or targeting TNF might actually have beneficial effects. So it could go in that direction. We're far from this. That would be the next step from the basic research of trying to understand what happens if you block selective molecules. It could be also that simply giving an antibody against these molecules and complexing these toxic molecules will have the same effect. So that's for the future. But clearly, at this stage, TPE is the best that we can do in terms of removing these toxins or these negative factors. And we've removed them globally. But what's interesting to note is we might also be removing some beneficial proteins at the same time. So like in anything in science, you start with a very broad observation, and then hopefully you will refine it to a much higher level of resolution, which will frankly increase the specificity. And this is where we are now.
So would you recommend someone that is eagerly would like to live longer to do TPA treatment today, or should he wait a few years to get a bit more clarification about what are the benefit and the risk of that?
Well, it's a very difficult question for me to answer because I don't want to be wearing my company hat and telling this audience that everybody should come and get TPE. There are a number of factors that go into making this decision. Number one, have you optimized your lifestyle? Maybe not perfectly. I mean, I'm not living perfectly, but I'm trying my best. And once you have reached that plateau, do you want more? That would be the type of patient that I would recommend would do this. Someone who feels, I want to put everything on my side. And I think this is another experiment that's showing promising results. And I think I'm willing to give it a try. And I will, for example, on an individual basis, you could measure all of your clocks before and after, do it and see if you see a response. I would encourage you to continue. It seems to be working for you. If you don't see any response and you don't feel any difference, that would be more questionable. By the way, we had some patients, and my experience working with people undergoing this type of therapy, some people have experienced really significant changes in the quality of their lives. Some others really couldn't tell. So it's going to be something that has to be decided by people on an individual basis in consultation with their doctors or their health advisors. What we've been able to do for the first time is to show that there's clearly a beneficial effect, statistically significant effect in people with some individual variation. So this is the best I can give for people to, who decide that they want to give it a try or not.
Yeah. So, Eric, thank you so much for that and for the discussion. And I know how busy you are, especially in those challenging days in the academia that grants are cutting, and I'm sure that you are worried about how can you recruit more funding for the back. And I wish you good luck. And I'm looking forward to exploring the research in the field of longevity each week with you and other scientists. For more information, please go to www.insidetracker.com/podcast. Again, thank you so much, Eric. That was a very interesting discussion, and I think that it's a very exciting domain, the TPE, and I'm sure that our audience will appreciate listening to this episode.
Thank you, Gil. I think I always also want to thank you. I think your company, Inside Tracker, is one of my to-go company. I recommend it to many people as a way to actually monitor your health. I think you're playing a critical role in this whole landscape. I also want to make a pitch to potential donors. I think this is indeed a time when the field of longevity research, which frankly has been grossly underfunded, is looking for people to come and help us. We have an incredible bottleneck between the discoveries that we have in the laboratory setting and our ability to conduct clinical trials. And you might not know this, but the BAC, we've built over the last 6 years a clinical research center that John Newman has been building. We now have, additional faculty such as Lee Hood and Nathan Price, I think, and David Furman. So we're building a real clinical infrastructure that I think is going to be critical in the future to really open up the bottleneck and allow us to test all of these promising interventions in human in a proven manner, using the best tools, using the standard of care, using the hardest criteria in terms of efficacy. And in my opinion, this is how the field is going to move forward.
Yeah, and I want to add to that, Eric, that I completely agree with you. And I think that the BAC is an amazing institute, and the contribution to the science and especially to the longevity science is very high. And what you have done in the last 9 years since you joined the BAC, you transform it completely. So I want to thank you, and I assume that in behalf of our listener, but also in behalf of the scientific community, what you have done for the BAC is amazing. And the BAC is basically the horse that pushing all the aging research in the world. Is the first and maybe the biggest institute of studying aging. So thank you so much for that. I really appreciate it. And I think that you deserve a lot of positive reinforcement for what you are doing. Thank you so much.
Thank you. This is a labor of love and passion, and I'm surrounded by an amazing faculty, employees, and we were moving forward and really hopefully change the course of humanity and healthcare. So thank you for having me.
Thank you.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out, using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.